right, good morning everyone. So thank you very much for the organizer to pick my abstract and chosen me for this short oral presentation. So today I'm going to share with you some of the work that I've done in collaboration with my collaborator from A-Star Skin Research Lab, Sophie. So later I'll share with you how we got together and work on some of this work we have done together. Right, so I think this audience doesn't need much introduction to nutraceuticals. So basically, they are biological active phytochemicals that possess a number of health benefits, and they are very important in terms of maintaining well-being and support the function of body. And you can see them in a lot of fruits and vegetables that our previous speakers have spoke about, and they can be derived from botanical or food byproducts. And the reason that we are interested in food byproducts is because of food wastage. And it's a growing problem globally that's affecting both industrialized and developing countries. Right, so I'm sure most of you that have been to supermarkets and you choose your fruit and vegetables. And we have this phenomenon called cosmetic filtering. And what we mean by that is, of course, if you look at ladyfinger or your okra, if we see both of these being sold, I'm sure most of you, or at least I can speak for myself and my wife, we can spend a lot of time picking out okra that looks like this from this basket of okras. And we know that based on statistics, almost 50% of the wastage are attributed to fruit and vegetable due to their short life span. And these are a good source of biological active phytochemicals. And so what this means is it represents a potential wasted source of high-value biomolecules which could be extracted or even upcycled into other functional products, which we are interested in looking into skin. And why we are interested in skin photoaging is because skin photoaging is a visible, lifelong, cumulative consequence of UV exposure. And you can see over here when we expose our skin to prolonged UV exposure, then we have wrinkles, spots, loss of elasticity in the skin that has significant impact in the psychosocial and economic impact. And most of the conventional care, we use sunscreen and topicals that focus on damage control. So this doesn't really address the ongoing systemic oxidative stress and inflammation, which I think previous speakers have also talked about. They are very important. And a lot of current nutraceuticals such as collagen, antioxidants, or even polyphenols are now put into our beauty products that you can see over here, and they are very good in supporting our UV defense and, of course, our skincare. And this is also an ongoing field because of our aging population and the demand for non-invasive prevention of driving rapid growth and of this basically skin-focused nutraceutical market. So based on this connective market research report, and shows that this field is growing at a rapid rate of 8.3% compound annual growth rate, and is projected of this market size of $10.4 billion by 2030. And that's how I managed to reach out to Sophie from the A-Star Skin Research Lab, who is the expert in photobiology. And she has an ongoing collaboration with P&G that is involved in doing the science behind developing this nicotinamide in some of the products you see on the shelf. So now we have a good source of food waste that's being discarded, and we know what is the target for that. So what is missing in between is a way to extract these bioactive molecules from some of the food products. And in my time, before I joined NUS, I worked with this colleague of mine, Dr. Ong, and he has this method of extraction, which is called pressurized hot water extraction. And if you look at the literature, it's also known as subcritical water extraction. So what is happening with this extraction method is a green chemistry extraction method that only uses water, and it does not even use a single drop of organic solvents. So this is a DIY setup in the lab where we have the sample being packed in the extraction cell, and then we just need to pressurize and pump water through the extraction cell, and then we can have an oven that can control the temperature that influence the optimization of the extract that is coming out at the end. So once the extraction process is over, we can freeze dry, then we have the food extract that we're interested in. And this method is quite versatile, and it's suitable for many sample matrix. So you can see we have published work, right, using extracts from botanicals or herbal preparation, and we have recently also looked at food products, okras, and we also look into unavoidable food waste, which is your fruits and seeds that we usually cannot consume. Right, so overall what we can do is we can grind and process these waste into fine powder, pack them into the extraction cell, then we can perform pressurized hot water extraction. We got the food extract, then we have to then identify using HP, LC, LCMS to determine what is the bioactive molecules that is present in the extract. Then we characterize their biological properties, and then we can test the extract in various models. So in this case, we are looking at okras and testing of that in the skin. So moving on, after we take our food products, okra, so you can see once we cut them out, we see various parts. So I'm interested to see what's present in the different parts of the okra. So we can separate the okra into the seeds, the outer skin, and the inner skin. So our main goal is to identify and look at the chemical fingerprinting of what phytochemicals are present in the extracts. So you can see using a PCA plot of the three different compositions. So you have inner skin, the outer skin, the seeds. So we observe that different parts of the okra, whether it's the skin, the seeds, and outer seeds, once extracted with our extraction method, have distinct chemical profiles. So we also look at what are some of the common polyphenolic compounds that can be detected from these different components. There we can see that we can actually have a good amount of different types of flavonoids and polyphenolic compounds. They are different in quantity between the outer skin, inner skin, and the seeds. So what we found is, of course, we have detected a high amount of flavonoids such as quercetin derivatives, such as quercetin, glucoglycols, and mineralosucrose, and camphor, and these are detected significantly higher in the seeds than the outer skin and the inner skin. And once we know what they have, and then we perform a free radical scavenging assay using the DPPH. So we perform a dose response curve of the extracts, and we can see that the seeds have a much higher antioxidant capacity compared to the outer and inner skin. And also look at the ABTS, vitamin C antioxidant capacity assay. This also shows consistent finding that the seed extracts has the highest amount of antioxidant activity compared to the outer and inner skin. What we also show that besides having antioxidant ability, they also have anti-inflammatory activity. So we show that using a TNF-alpha induced endothelial inflammation model, so okra seed can also reduce the expression of inflammatory markers such as VCAM1 expression. So once we saw that happening in our lab, then we collaborated with Sophie's lab, where we can test the potential anti-aging effects of these okra seeds in vitro skin models, such as using the human primary keratinocytes and 3D organotopic models under photoaging conditions. So in Sophie's lab, they perform a series of testing, firstly using 2D human primary keratinocytes. So they seed the primary cells, and then they can irradiate the cells with UVA and UVB to damage the cells, and then they replace the media, and then that's where okra seed is being treated for 3 days, and then they harvest the cells to look at gene expression of various inflammatory markers and differentiation markers. So firstly, we look at some of the inflammatory markers, PTGS, ICAM, and IL-8. So you can see that UV exposure significantly increase these inflammatory markers, and co-treatment with our okra seed extract can significantly reduce these inflammatory markers. And nicotinamide is also used as a positive control in experiments that we can see they are definitely working similarly with our okra seed extracts. Next, we also look at differentiation markers, filaggrin and involucrin. So you can see that similarly UV increases these late differentiation markers. So suggesting that they are prematurely aged, and then with our treatment with okra seed and the positive control, so they can significantly downregulate the differentiation markers. And moving on to the 3D organotopic models, so you can see that these are 3D cells, so they can be inflated into a 3D skin block. So then we can treat the okra seeds and then irradiate, and then after 8 days or so, then they can harvest the tissue for histological analysis, and measurements of cytokine also shows that UV increases inflammatory markers IL-6, and this is significantly reduced. And also looking at premature differentiation using laminin B1 as a marker, we can see that this is also reduced in this condition where it is treated with okra seed. And just to summarize, so what we have shown over here is we have an extraction method that is green, that doesn't use organic solvent, and we can be suitable for a range of different food byproducts. So we can extract them, test what they are present, and we can perform basic antioxidant and anti-inflammatory tests. Then we can go into efficacy testing in the skin models, and then we showed that they are capable of reducing UV-induced differentiation and senescence in our 2D and 3D models, and this shows that these okra seeds have anti-inflammatory activities and anti-photoaging. And what we plan to do in the future is, since I'm in engineering, so we're also looking into digital fabrication using 3D printing. So some of this extract may be fed with hydrogel or microneedles to enhance its drug release.
Yeah. Okay, can we have the microphone back? So this is, yes, for not more than 30 seconds.
No, it's the last slide.
Yours last slide. Okay,
Thank
good.
you.
Thank you so much.